Lifeline?
No, they gave me bad.
Bad advice?
Well, man, I'm sorry to hear that.
All right, everybody. So let's talk about these. 7, about somebody growing cells that don't have any lysosomes. And the problems, right? So again, when I want you to think about the function and form of these organelles and other types of cell components, you should also be thinking about, well, if you know that, if you can recall this component does this action, then sort of reverse it and think about, well, what would be the deleterious, what would be the negative effects if the cell did not possess that particular organelle? And you should be able to relate that back to its function, right? So no lysosomes. All right, what's the purpose of the lysosome? You think about the function of the lysosome, it's about degrading waste, getting rid of bad stuff, breaking it down, getting it out of there. So any answer there that might be tied to the fact that the cytoplasm would then probably become toxic, that there could be issues with maintaining homeostasis in the cell. There would be an accumulation of waste products that could potentially inhibit metabolism or inhibit gene expression, transcription, translation. A multitude of things could happen if you are trying to, if there's waste where there shouldn't be waste and if it's accumulating. So that would sort of be the effect. These cells would probably not last very long because they'd basically be poisoning themselves to death, right? It'd basically be like walking around with a plastic cover over your head while the carbon dioxide takes its toll. That was pretty morbid. Anyway, compare and contrast cilia and flagella. Hopefully that was pretty easy. I think I took that one verbatim from the end of the chapter. So flagella tied to movement, cilia tied to movement. They're both exterior. They're both anchored in the plasma membrane of the cell, or the cell wall, if it's a bacterium, for instance. In terms of differences, you could talk about their relative length. Cilia are very short. Flagella are very long. Cilia also might have other functions that are tied to attachment. So anything along those lines? All right. All right, so we had a question about tight junctions and gap junctions, right? So it would make sense if our intestines have tight junctions, then probably be good for skin to have tight junctions too, right? So if it had gap junctions instead, what does that mean? Well, it would mean that your skin cells, the living layers of your skin cells, it would be easier to transmit between them, I suppose. But then if you're thinking about, well, if you no longer have tight junctions, there's going to be space in between those cells, right? They're not fused together anymore, where the cytoplasms come together. So if you think about that in terms of the skin's going to become more permeable at that point, right? Why is that a bad thing? What's that?
Any type of germ or bacteria in the air penetrate your skin.
Right, or rain or water or liquid. There would be a greater capacity for your skin for potentially toxic chemicals or organisms to penetrate the skin much more easily. Okay? Now you might still be somewhat okay because skin has other properties that prevent those things from happening. Right? They're waterproof. Imagine if our skin wasn't waterproof, the potential issues we might have. But nevertheless, I think that would be sort of the manifestation if junctions got swapped with respect to the skin.
Does that impact pain threshold? So like your sensitive skin, if the junctions?
Right. So that I do not know. It's a good question. Does anybody know? I'll look it up. It's be on record. So I have to look it up because I said I would. All right, so let's see if there's any type of correlation between pain tolerance or pain sensitivity, the converse, and the form of junctions in the skin. Yes?
From somebody who hasn't really researched this, it sounds like it would affect it because then the signals could get to the nerves more easily,
Uh-huh.
and then the nerves would feel the pain, but that's just...
That's possible. That's possible. Or it could be anatomical in the way in which maybe some people have more layers than others. Maybe the strength of the signal is genetic. I don't know. I'll check it out. See if I can find something. I'll ask Dr. Leonard. She'll know. All right. White blood cells. This one was pretty hard, I admit. All right. Would you expect white blood cells to contain more microfilaments than red blood cells? I would say yes, you would. Now, I could be convinced if you said no, so I'll read through these and see. But why would I say yes? So what do microfilaments do? If you had a cell with a lot of microfilaments, what would that allow them to do, potentially? Change shape? Is that? Change shape, move, migrate, right? Does microfilaments contain actin, which is going to allow that type of movement? So what are some things white blood cells need to do? You cut your finger, it starts to swell up. That exudate starts to form, right? What's happening? White blood cells called neutrophils are migrating to that. They pick up that stimulus and they're migrating to that region. They're escaping the lymph. So they're going to have to be able to move themselves in order to get to there. And then when they get there and they find some bacteria that's got into that wound, they phagocytize it, right? They're going to engulf it. So that's going to require movement. They basically have to extend themselves and grab the foreign material, the antigenic material, so that it can be digested. Red blood cells, do they really have that type of function? Think about what state red blood cells pretty much exist in, right? They're flowing through the bloodstream. There's already a current, right, which is facilitated by the smooth muscles of the blood vessel walls in addition to the pump, which is the heart. And therefore, I would argue that white blood cells, because of that, you would expect them to have more microfilaments as opposed to red blood cells. Does that line of logic make sense? All right. If you put no, I'll read it, and if that makes sense too, then you're good to go. All right, 11. One cell component universal to all cells, and explain why. All cells have, DNA, obviously, genetic material in order to divide. Could be an answer. Plasma membrane. Ribosomes. Could be an answer. All right, and then what would be universal? So the bacterial cell one is probably the hardest one to think about. So, does anybody have an answer that they think is that they got it right? You all?
You put nucleoid, because other cells have a membrane bound nucleus.
That can count. So bacteria have this nucleoid. So it's this slightly different arrangement of how it deals with its chromosome or chromosomes. Some bacteria have more than one, not very many, but nevertheless, that could be counted. Did you all have something else? Okay, nucleoid. That's the thing that's probably out of this chapter would be the easiest thing to describe. There's other components that the chapter doesn't get into that bacterial cells might have. For instance, bacterial cells sometimes have circular chromosomes as opposed to linear. You could argue that, as well as some really minute things that exist. But I think nucleoid makes the most sense then. Animal cells. What are some examples in animal cells? What'd you all put? Centrosomes. Okay. Anybody else? Lysosomes. Okay. What about plant cells?
So.
Cell wall, you gotta watch out for
Sorry,
cell walls,
chloroplast.
right? Because bacteria have cell walls, right?
Chloroplast and chlorophyll.
Right. Plant cells, think about the act of photosynthesis and the mechanisms by which it produces its own food, which would be chloroplast would make the most sense there. Yes?
So I know that animal cells have vacuoles too, but we put large central vacuole, but that's so big.
Yeah. Right. So, again, there's that list in the book, there's that table in the book that sort of differentiates prokaryotes, prokaryotes, animal cells, plant cells. That would be a good thing for reference as we get closer and closer to the examination to become familiar with in order to be able to properly describe any of the differences that might be known when comparing those three groups. All right, so sounds like you all did pretty good on this. Happy to hear it. So for our last piece of this, I'm going to, let me go grab the exams and I'm going to talk a little bit about it before we do it. All right, so let me first talk about the exam itself and the format and what you're going to see when you get your exam, and then also talk a little bit about just reflecting on what you're going to see and perhaps some strategies or support that I want to try to provide or that the university can provide if you want to get a better score next time, and sort of go from there. So first of all, the exam itself, as you may recall, hopefully, so it's 3 sections. What you're going to see in your exam is, I'm going to give back your exam, your actual physical exam, so that's going to include your answers for the short answer, section 1. So you're welcome to look at that. I will need that copy back today. So you can take pictures of it if you want, of those pages, but really just the first 3 pages are most pertinent. You'll also get a score sheet, which will let you know how the Scantron read your exam, so that pertains to sections 2 and 3, the matching and the short answer. So you can take a look at that. And then you'll also receive a little script that reflects your score and what you scored on each section and your total score. Okay? Remember, the exams are 15 points, so 15% of your total grade. It's not that extensive. But nevertheless, you'll get to see how many points you accrued and what your percentage is. When I get back in my office, I'm going to flip a switch, and then you'll be able to see that same score, hopefully, in Moodle, if I did it correctly. So, again, as I anticipated, the recall sections was the section that most people did the best in, and then followed by the understanding section, and then the application section. But again, these are tiered. So if you did really well in the recall section, that boosted your score a lot because that was worth the most. That carried the highest weight. Okay. I want to implore anybody that feels discouraged based upon what they see on their score to do the following things. Number 1, come talk to me. If you scored a certain grade, I will contact you just by email, just saying, hey, what's up? Is there anything I can do personally to help you out? I will do that. Of course, there's tutors available for free that your tuition pays for that should help you with this, so I can mediate or facilitate that to happen if that's going to help. For those of you who get your exams back and actually and feel like you were successful, I'll be sending out a message later on. I would really like to hear your input on how you think you were able to accomplish that, in the hopes that perhaps that can help other students. So if you studied in a particular way that you thought really helped, if you did something that perhaps others might benefit from, I would really like to hear your feedback there, so I'll send a link so that you can submit those anonymously, and I'll share them with the group when we think about the next couple exams. Hopefully if you watched the video I made, it looked like some of you actually did, hopefully that helped sort of that helped you figure out the way in which you'd be best prepared for this. I basically took every single outcome from each chapter and made sure there was at least one question there and actually tried to sort of make it equal across all of those. Okay, so I think that's pretty much my spiel. So again, when I pass this out and give it back, you can keep the little strip you're going to get with your percentage. You can keep your score sheet. You can take a look at your actual physical exam. But before you leave, I will need to collect it back. Was there a question? Yes?
Do you on the top of your head know the class average?
I do.
Oh, God.
Class average is basically almost straight 70%.
Oh.
All right? So the exam key will be posted immediately after this class. So if you want to look back, again, if you don't want to take pictures or anything, all of that with answers will be posted on Moodle in a particular section. Okay? So the mean and the median were both around 70%, which means that, you know, that gives you a sense of sort of where everything was. All right? Yes? Do I curve my test? I do actually look at the percentage. So if there was a particular question that was incredibly difficult, meaning that less than 20% of the students got it right, what I do is that I'll add a point there. So if you got it right, you got a really hard question right, you still are benefit. But if you got it wrong, I throw it out. So that only happened about maybe 2 questions on this exam, I think, 2 or 3. So by and large, the majority of you got most of the questions right. I only do that for the Scantron questions, not the short answer. In terms of the short answer questions, let me also mention real quick, I basically use a 3-point scale on how I assess those. So 0, you know what 0 means. If I think the question was answered to some degree of adequacy, you get 1.5 points on that. If I think it was a really good answer, you get 2 points. Okay? So that's sort of the scale that I use. That's the same scale I use on the CATs as well. All right, so start sending these out. Take a look as long as you want, and make sure you give back the exams to me. Metabolism chapter will be next. I plan on having all that content uploaded to Moodle. If not by today, by first thing tomorrow morning. All right? RAT will be due like it normally is, end of Monday, September 18th. All right. Samantha. It might be easier, yeah, if I call your name, you can come get it. Ollie? Ashley? Ashley down here? Kendall? Oh. Ed? Catherine? Oh, I'm sorry. Sorry about that. Gave you the wrong one. Catherine? Hillary? Tamara? Grace? Cortisha? Brittany? Raven? Cole? Elise? Whoops. Sorry about that. Henry? Manella? Rachel? Hannah? Kelsey? Lily? Devin? Sarah? Whitney?